They're both monocots. What's the difference? The difference is the world has given you maize that you're eating. The world has given you also this grass, but you're not eating it. But it can survive drought. What we do is we find out and ask the hard questions. Why is this grass, which is also maize, which is equally a grass, if you allow me to loosely call them that, is able to survive? That is where we come in. And that is why I just give you a very brief definition of agricultural biotechnology, which simply means it is the application of the scientific techniques to modify plants. Remember, modify plant, animals, microorganisms used in agriculture to improve their traits and enhance productivity. Very important. Now, if there is nothing else you come out of this place, editors, please go away with this slide. This is the only slide that is useful. The basic knowledge here is that if anything that is alive that is walking out there, be it a plant, be it an insect, all these things that are living, they all have something in their cells at the central part of it called the nucleus, which is called DNA, or nucleic acid to be more precise. Because DNA is just one shade. There is also RNA, which is also the same thing, but allow me to call them nucleic acids. I have a background in biochemistry, so that's where we deal with that. Nucleic acids, that is DNA or RNA, but you hear DNA more than you hear RNA, realistically so. In that DNA, in that nucleus, there are the things that we are calling DNA. The DNA of anything that is alive, and please listen to me, only have 4 letters. 4 letters. It is the A that you're seeing there, it is the T, the G, and the C. Those are the 4 letters that repeat themselves. So if you look at the photos there, you'll see the question would be, why is the elephant then different from the horse and from penguin and all that, if they are only 4? Good question. But now the variation is that those 4 letters repeat themselves in that DNA in a particular sequence. So the variation is the sequence of repetition and the size of the content. For instance, for the human beings, you and I, in our DNA, in our nucleus, we have 3.2 billion of those ATGC or whatever repeating themselves in that genome. It has been sequenced. Now, that is where now it becomes very interesting. Look at the DNA or a cell as an organization. You are editors. Your business is in news, whether in print, TV, or radio, all those things. If you look at the cell as an organization or a human being as an organization, then the headquarters of this NTV, which is here probably in Nation House, is the nucleus. And within that headquarters, we have software, the documents that are used to run that organization. For instance, pick the HR manual. That is where everything else happens. If you want to employ, where do you go? HR manual, what does it say? When you want to promote, when you want to sack, HR manual. So it means every time you go back to the HR manual. That is what the cell also does. If there is drought, the DNA is asked, what do we do? If there is rain, too much of it, they ask, what do we do?